I have test 3 with me. I'm going to hand back test 3. So those of you joining by Zoom, just stand by, bear with me for a little bit until this is done. Okay, Kelsey's joining by Zoom today, so she's not here. Bethany, I don't see. Those are the BIOLs. Is Jacob Bishop here? Joan? Clear, Fire Collins? Seth Rozier? Here, Chris Ray. Here, are we guys not see? It'll be in before your final grade. There is just I have a lot on my plate right now. I have 2 classes all of COVID procedures. There's a lot of special accommodations for the grades. But yeah, it's, I'll be working on a lot of them. Hopefully, the lead. Both hiring and money. Joel and Ashton and Kyle. Okay. Is there anybody here today who did not get their test yet? Okay, so again, I'm not going to go over them because all of your answers are in the student grade report on the last page. So if you got one wrong, you can write the right answer the first student grade report. And then, of course, you can get a study for the final exam coming up on Monday. Okay. Oh, one more announcement. They had asked me, is there any way you can post test 3 so if you didn't come in, see, get it back, you can still use it to study for the final? I could do that. I'm already testing, going to post test 2 because it's lost. Claire wants to come in and test around 1 if I'm in. I'm not planning on being in, but I'll tell you what, I'll stick around. So if anybody wants to, if anybody who's joining by Zoom, 1 o'clock would be a good time today to come by. and pick up your tests. Okay, one more thing before we continue with the chapter on the nervous system. So remember that we're doing quizzes every day this week. So what I'm going to do today, I'm actually going to post 3 quizzes. Okay, I'm going to post 1 quiz on what we covered today, and the other 2 will be on just random things that we've covered since test number 3. Okay. The reason I'm doing this is because I want to get to at least 24 quizzes so that everybody has at least 4 drops. All right. So remember, the way this works is that I only count your 20 highest quiz grades. So if you take all 24 quizzes, that means your 4 lowest grades will be dropped. Now, if you don't like the idea of having 3 quizzes over the weekend and you don't want to take them, then don't take them. Okay. Still only your 20 highest quiz grades are going to count. But for those of you who really want to maximize your points from quizzes, I would suggest taking as many quizzes. The more quizzes, the better. The more quizzes we do, the more drops you get. So I would recommend taking all of the quizzes. Okay, and I think that's about it. And actually, Colton, I realize we haven't taken care of your that makeup. I'll take care of that later today as well. All right. So I think that takes care of everybody for the time being. So let's continue with the chapter on the nervous system. So when we left off, we were talking about an action potential. And an action potential is the electrical impulse that travels along neurons. So this is important in communication, so signals going from the brain to the tissues, or vice versa, going from sensory neurons back to the brain. So the way that this happens, remember that the cell membrane is polarized, okay, because of this protein called the sodium-potassium pump. So the sodium-potassium pump maintains the resting membrane potential by pumping sodium out of the cell and potassium into the cell. This makes for a concentration gradient, well, I should say concentration gradients across the cell membrane. So we have a higher concentration of sodium outside the cell, lower concentration inside the cell, and conversely, we have a higher concentration of potassium inside the cell and a lower concentration of potassium outside the cell. Okay? Also, because 3 potassium are pumped out for every 2, 3 sodium are pumped out for every 2 potassium pumped in, this also sets up an electrochemical gradient. So the inside of the cell is negatively charged with respect to the outside of the cell. Okay? So this is what we call resting membrane potential. So we have a high concentration of sodium outside, high concentration of potassium inside, and we have an electrochemical gradient that's about -70 millivolts at resting potential. Okay? Now, when an action potential happens, the cell membrane, or part of the cell membrane along the axon, gets depolarized. So sodium ion channels open up. When sodium ion channels open up, sodium ions rush in to the cell. Okay. Now, this is going to depolarize a small region of the cell membrane. So after a short delay, potassium ions rush out of the cell. So potassium ions channels will open up after a short delay. When the potassium ions rush out of the cell, this repolarizes the cell. Okay? Now, in the meantime, the sodium ions are going to diffuse outwards, and they're going to cause more sodium ion channels to open. So remember that there's several different ways we can gate these channels. Voltage gated is one. So sodium ion channels are going to open up in response to depolarization of the plasma membrane. This depolarization is going to cause more sodium ions in the channels to open. That's going to further depolarize the plasma membrane. Okay, then again, after a short delay, potassium ion channels open up. Potassium rushes out. That repolarizes the membrane. And so there's going to be like a wave-like depolarization of the membrane that proceeds down the axon of the neuron. Okay, let's have a look at the next slide. This might make more sense. So this is what happens during an action potential. So over here, we're at resting membrane potential. This really messes up when I try to write on it. Let's just put RMP for resting membrane potential. So resting membrane potential is about 70 millivolts. Here it's 60. So 60, 70 millivolts, that's right about where it should be. All right. Now remember that sodium ion channels are going to open in response to certain stimuli. The stimulus can be from sensory neurons, for touch, taste, light, smell, something like that. Or other neurons can also stimulate sodium ion channels for neurons to open up. All right. However, it has to get past a certain threshold before an action potential is triggered. Okay, so over here, so we can see at resting membrane potential, the sodium ion channels are closed and the potassium ion channels are closed. Okay. Now, so in response to a stimulus, we can have the opening of some sodium ion channels. Okay. This is going to cause sodium. Remember, we have a high concentration outside the cell, low concentration inside the cell. This is going to cause sodium ions to rush in. Okay. When sodium ions rush in, that is going to depolarize the membrane. So we're going to have a localized depolarization of the membrane. Now, if that depolarization reaches a certain threshold, that's going to trigger the membrane potential, the action potential. So over here, if it gets above a certain threshold, it's going to cause more voltage-gated sodium ion channels to open, and more sodium ions are going to rush in. That's going to depolarize the membrane, which is what's happening here. So sodium ions rush in. That causes the membrane potential to go from -60 or -70 all the way up to about +30 or +40 up here. All right. Now, after this happens, there's a slight delay, and then the potassium ion channels over here. Okay, the potassium ions are in pink. This causes potassium ions to rush out. The rushing out of the potassium ions is going to repolarize the plasma membrane, which is what's happening here. Okay, so what happens is the potassium ions repolarize the membrane down to about -60 or -70. Then they actually repolarize it even more. So they kind of overshoot the repolarization. Okay. Then over here, the sodium-potassium pump, remember this is that protein that pumps 3 sodiums out, 2 potassiums in each time it works. So the sodium-potassium pump is going to restore the normal concentration of sodium and potassium and the normal resting membrane potential. So that's what's happening here. Okay. So again, over here we have resting membrane potential. If enough sodium ion channels open by stimulation, that it goes above a certain threshold, that's going to cause depolarization here. Okay, that's when these voltage-gated sodium ion channels open up in response to a drop in voltage. Okay, that drop in voltage is going to cause more sodium ion channels to open up. Okay, and if that, again, sodium ions are going to rush in, that's going to cause more sodium ion channels to open up. So it's like a chain reaction. It's gonna happen like in a wave all the way down the axon of the neuron. Okay. Then after a slight delay, the potassium ion channels open up, which is what's happening here. Okay, that's going to repolarize the membrane back to -60 or -70, kind of overshoot it a little bit. Then here, from here, oops, that annoys me. So anyway, from here to here, this is where the sodium potassium pump restores the resting membrane potential of the neuron. I know that's a lot. Is everybody kind of following that? Good. Well, I'll tell you what. Well, let's talk about it some more. I'm going to show you that video again because that is a pretty good video that shows how things happen. But let's go over that again on this slide. So the action potential starts in the trigger zone and propagates down the length of the axon. Okay, so again, it's going to happen kind of in a wave-like manner down the length of the axon. The depolarization at the trigger zone allows sodium ions into the cell. So the trigger zone, this is at the beginning of the axon, right near the cell body. Let's go back and take a look. Okay, so looking at a neuron, the trigger zone is going to be here. Well, we can't see the whole neuron. Let's go back to the better. Okay, so let's concentrate on this and this. So the trigger zone is going to be right here near the cell body. All right. Now, again, the trigger zone, this is where if the membranes depolarize past a certain threshold, that's going to trigger the action potential. Okay, and again, we're going to get a wave-like depolarization of the plasma membrane that's going to proceed down the axon. Okay? And it's always going to go in this direction. Okay? It's always going to travel from the cell body towards the axon termini in this direction. Okay, so again, the depolarization of the trigger zone allows sodium ions into the cell. This is going to depolarize the membrane. The sodium diffuses down the axon to the next set of gates. All right, so remember that these sodium ion gates or these sodium ion channels are voltage gated, meaning that they're going to open up in response to depolarization of the membrane. This raises the voltage at the gates, again, so that, again, it's going to depolarize the membrane. And these other sodium ion channels are going to open in response to that depolarization. Then more sodium is going to diffuse to the next gates, further depolarizing the membrane, and that's going to trigger more sodium ion channels. Okay, so this is how it's kind of a chain reaction of opening sodium channels going down the axon, because these are voltage-gated channels, and opening them depolarizes the membrane. This continues all the way down the length of the axon until it reaches the axon terminal. The wave of depolarization followed by repolarization down the axon is known as the nerve impulse. In the refractory period, the potassium gates close slowly, so the membrane potential drops down below -70 millivolts. It takes time for the neuron to return to -70 millivolts, and that's because the sodium potassium pump has to work to restore the normal concentrations of sodium ion, potassium ion, and the normal electrochemical gradient. The neuron cannot fire another action potential for a short period of time. So until the sodium potassium pump restores the normal concentrations of sodium ion outside, high concentration of sodium ion outside, high concentration of potassium ion inside, and this -70 millivolts electrochemical gradient, the neuron can't fire. So that's what the refractory period is. It's the period where we're restoring the normal resting membrane potential, and the neuron actually can't work all the way. Okay, so now I want to go back and show that short YouTube video again because I think it might make a little more sense to you now that I've had some time to explain this a little bit better. So let's stop the share. And okay, now I showed this, I know I showed this to you on Wednesday, but I'm thinking now that we went over this more, it might make a little more sense to you. So let's have another look.
That is not transmitting nerve signals. What happens when the cell is stimulated? A stimulus changes the permeability of the cell membrane to sodium and potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal called an action potential that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close depending on voltage changes across the membrane. When no nerve signals are being transmitted, these channels are closed. A stimulus causes voltage-gated sodium channels to open, and sodium ions rush into the cell. The cell becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close, while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The cell returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in, and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. In the wake of the action potential, potassium leaves the cell, restoring a negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, reestablishing the rest of the potential distribution of sodium and potassium ions. Let's look at another action potential as it moves along the membrane. This time we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal of membrane polarity. Sodium channels close and potassium channels open. As potassium ions rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.
Right. So I'll post the link to that on YouTube. Please let me know if I forget. I know I have an awful lot of work to do. I need not just this class, but other classes too. So I might forget something. So if I do forget about that, please email me and let me know that I haven't done that. Let's go back to the PowerPoint. All right. So before we continue, I just want to make sure everybody's okay with how action potentials work. I know if you have to think about it a little bit to kind of get your head around it, but everybody understand how that works, more or less? Okay. So next, let's talk about the speed of a nerve impulse. In invertebrates, the speed of the action potential down the axon depends on the diameter of the axon. In narrow axons, action potentials travel a few centimeters per second. In the giant axons of larger invertebrates, the action potential can travel as fast as 30 meters per second. All right. Now, they talk about the giant axons of larger invertebrates. When scientists first started studying neurons and how all of this stuff works, they did it on the giant axons of squid. So this is what the slide is referring to when they talk about the giant axons of invertebrates. So for whatever reasons, squids have these enormous neurons. Okay, they're quite long, and because they're so big, they're very good. They make very good subjects for study of the nervous system. Okay, so we learned an awful lot from giant squid neurons. In vertebrates, the speed of the action potential is increased by insulating the axon with myelin sheaths. So remember when I talked about myelin sheaths before? Myelin sheaths are made from these special types of neuroglial cells called Schwann cells. And these act very much in the same way that insulation works on a copper wire. Okay, so in vertebrates, the speed of the action potential is increased by insulating the axon with myelin sheaths. Glia cells wrap around the axon, forming the myelin sheaths. Between the cells are unmyelinated areas called nodes of Ranvier. Action potentials jump from node to node, speeding up the transmission. Fewer action potentials needed. Human axon is 20 micrometers in diameter and has a transmission speed 40 times faster than that of the giant squid axon. Okay, so in invertebrates, when they don't have the myelin sheath, the speed of the transmission depends on the thickness of the axon. Okay, more thicker axons, faster. In humans, even though the axons aren't particularly thick, not as thick as those of the giant squid, the myelin sheath is what increases the speed so that it's even faster than that of a giant squid. Okay, so a synapse. Synapse occurs when the axon ends and transmits the message to the next cell. Okay, so I mentioned that neurons can be stimulated by other neurons, right? So the dendrites of one neuron can synapse with the axon endings of another neuron. Okay, now what's a synapse? A synapse is a very small space between a neuron and another cell. Let me write that down on the board. So a synapse this is a very small space between a neuron, I should say a very small space between the axon termini of a neuron and another cell. Okay, so now, again, I apologize for my artwork because I'm no great artist. Okay, so this is supposed to be a neuron anyway. So we have cell body, dendrites, axon, and at the end we have the axon termini. Okay, so I mentioned that neurons can be stimulated by other neurons. So the axon termini of one neuron can interact with the dendrites of another neuron. And the small space between the axon ending of this neuron and the dendrite of this neuron, this is called a synapse. Okay, so we can have synapses between two neurons. We can also have synapses between muscle fibers and motor neurons. So again, in that case, the axon terminus of one of the neurons is going to associate with a muscle fiber, and there's going to be this very small space in between the muscle fiber and the axon ending. That's the neuron. That's the synapse, right? Let's go back to the PowerPoint. Okay, so a synapse occurs when the axon ends and transmits the message to the next cell. Usually another neuron's dendrite, but it may also be a muscle cell. It may also be a gland, so that electrical impulse causes a hormone to be released from the gland. These come in two types. There's an electrical synapse. Electrical synapses are rare, mostly found in the brain and the spinal cord. Ions or chemical messengers flow between the two cells through gap junctions. Hopefully everybody remembers gap junctions. Gap junctions are this type of cell junction that connects two cells. Again, gap junctions, we have a lot of gap junctions in cardiac muscle because it allows for communication. Turns out these gap junctions also allow for communication between cells of the nervous system in some cases. These are rather rare. This allows for rapid transmission in both directions. Most of these synapses, however, are going to be chemical synapses. Chemical synapses is where special chemicals called neurotransmitters move between the cells. Okay, let's go back to the whiteboard for just a second. Okay, so here we have two neurons that are connected by a synapse. So an impulse can be passed from this neuron to this neuron through that synapse. So let's just say that this neuron is stimulated. The stimulation is going to cause opening of sodium ion channels. That's going to cause depolarization of the membrane. Let's just say that stimulation is great enough to trigger an action. Okay, so then we're going to get this wave-like polarization that's going to travel down the synapse until it gets to the travel down the axon until it gets to the axon termini. Now, in a chemical synapse, that electrical impulse can't pass directly to the next neuron. Okay, it needs to pass in the form of chemical messengers called neurotransmitters. So when the electrical impulse reaches the end of this neuron, that electrical impulse is going to trigger the release of neurotransmitters into this small space between the axon ending and the dendrite of the next neuron. Again, the small space is called the synapse. There are receptors for those neurotransmitters on the dendrite, okay, and once those receptors are bound, it's going to cause channels to open up. Okay, again, that's going to cause more sodium ion to rush in. And if there's sufficient stimulation of this neuron from this neuron, where it gets above that threshold, that could cause another action potential to trigger in this neuron. So again, we'll have chemical transmission of the impulse down the axon. And then when it gets to the axon ending, if there's another neuron over here catching this, we're going to get chemical communication of that across the synapse. Okay? Does that make sense to everybody? So yeah, so the signal travels as an electrical impulse down the axon, but as a chemical signal through these chemical synapses where the neurotransmitters travel by diffusion. Okay, so still on the subject of the chemical synapse, the presynaptic cell is the one where the action potential is traveling down it. So on the whiteboard, that would be the one I drew on the left-hand side. Ends at an axon terminal. Axon terminal contains vesicles holding neurotransmitters. The cell membrane has voltage-gated potassium ion channel. Pardon me, voltage-gated sodium ion channels. Okay, so involved in the action potential, I talked about concentrations of sodium ion and potassium ion. Okay. Now, the concentration of calcium ion is similar to the concentration of sodium ion in that it's higher outside the cell and lower inside the cell. Okay. However, at the axon termini, these voltage-gated calcium ion channels are important because when calcium rushes into the cell, that's going to cause exocytosis of these vesicles so that the neurotransmitters are released into the chemical synapse. Okay. So again, remember that the presynaptic cell is the one that has the action potential traveling down. Okay. So again, the action potential is going to go, so as this depolarizes in a wave-like fashion, depolarization causes opening of sodium ion channels. But then when we get to the axon terminus, we have voltage-gated calcium ion channels. So change in membrane potential causes calcium ion to rush into the cell. The calcium ion is the signal for these vesicles carrying the neurotransmitters to fuse with the plasma membrane and release the neurotransmitters into the synapse, which is what's happening here. The postsynaptic cell is the one receiving the signal. Okay, so these neurotransmitters are going to bind to channels on the postsynaptic cell. When these channels on the postsynaptic cell bind to the neurotransmitters, that's going to cause these channels to open up. Okay, so again, the potassium ion channels were voltage gated, meaning they open up in response to a change in voltage. The potassium ion channels were voltage gated, meaning they open up in response to a change in voltage. The calcium ion channels were gated, voltage gated as well, meaning they open up in response to a change in voltage. These are what's called ligand gated channels. So ligand gated channels open up in response to binding of a certain type of molecule called the ligand. Okay. In this case, the neurotransmitter is the ligand. So when the neurotransmitter binds to these channels, that's going to cause them to open up. And again, this is going to cause sodium ions to rush in. That's going to depolarize the membrane somewhat. Okay. If we get enough depolarization, then above that certain threshold, then it can trigger the action potential, and we can get a second action potential. Is that making sense to everybody? I hope. Okay, the action potential travels down the axon terminal. This raises the voltage in the axon terminal, and the calcium ion channels open. Again, this is because these calcium ion channels are voltage gated. Calcium ion rushes into the terminal. Calcium ion causes the vesicles to move to the cell membrane, fuse with the cell membrane, and deposit the neurotransmitters into the synapse. The neurotransmitter diffuses across the cleft into the other cell. Okay, so that's one reason why these synapses, I mentioned the synapses are very small spaces. You may remember from lab 4, I believe, diffusion is really only efficient over very small spaces. Okay, so the synapse, and in the synapse, chemical communication is taking place by diffusion. So the smaller that synapse is, the more efficiently that the chemical communication is going to happen. Okay, so these need to be very, very small spaces. So the neurotransmitter diffuses across the synapse, and then of course binds to ligand-gated channels on the postsynaptic cell. Okay, and again, I went over this, so I'll just read over this quickly. The neurotransmitter binds to a receptor on the membrane of the postsynaptic cell. The neurotransmitter binds to a potassium gate. It opens and allows potassium out of the cell. Again, this lowers the voltage of the cell, hyperpolarizes the cell, makes the neuron less likely to fire at an action potential. So this is actually, in addition to stimulating the cell, we can desensitize the cell as well. So if these neurotransmitters bind potassium ion channels, they're going to desensitize the cell, okay, making it less likely that an action potential can be triggered. Okay? If the neurotransmitter binds to a sodium ion channel, okay, now sodium ion is going to rush in, that's going to depolarize the membrane, and that's going to make it more likely that an action potential will be triggered. Okay, so still on the subject of chemical synapses, a single presynaptic neuron may form connections with many postsynaptic neurons. Okay, so notice that we have lots of, if we look back at the whiteboard, we have lots of dendrites and we have lots of axon termini. So one neuron can connect to many other neurons to form a neural network, you know, or a network of these neuron cells. So a single presynaptic neuron may form connections with many synaptic neurons. So its axon termini can be interacting with the dendrites of many other neurons. It may simultaneously inhibit some cells while stimulating others. So what this means is some of those neurons that the presynaptic neuron is interacting with, in some of them, the potassium ion channels are going to open up, and it's going to make firing of the action potential in that neuron less likely. In some of them, the sodium ion channels are going to open up. Okay, so that's going to make an action potential in the postsynaptic neuron more likely. Whether a neuron fires an action potential depends on the sum of all the signals it receives at the dendrites. If the cell is receiving more IPSPs than EPSPs, the cell will not fire. So in other words, that cell is being desensitized or it's being inhibited from firing. If the cell is receiving the same number of both, it will not fire. If the cell is receiving only slightly more EPSPs and IPSPs, the voltage will not reach the threshold and it will not fire. If the cell is receiving many more EPSPs and IPSPs, it will fire. Okay, So this sets the bar pretty high as far as how much the neuron has to be stimulated before it's actually going to, before natural potential can be triggered and propagate down the axon of the cell. Okay, we only have 3 minutes left in the whole semester, so I don't think that's enough time to start another topic like neurotransmitters. So why don't we go ahead and stop here? Again, let me remind you, I plan on having some quizzes available by about noon today. I'm going to post 3. Okay, one's going to be on what we covered today. The other 2 are just going to be on topics that we've covered since test number 3. The reason I'm giving you 3 this weekend is so that we can get the number of drop low grades up to 4. So again, if you don't feel like doing quizzes and you're happy with your quiz points the way they are, of course you won't have to do the quizzes. But I would encourage you to do them to try to maximize the points you get from quizzes this semester. I will be in the office until about 1 today. If you want to stop by and pick up any tests. Again, old tests are really your best study guide in studying for the final exam. I haven't heard back from Biology Club about extra credit yet, so as to when that will be posted, I'm waiting on the Biology Club to contact me. But again, if you do make donations, make sure you record your name somewhere so that you get credit for it. Somebody's asking, I may have accidentally missed this, where's my office? My office is LSB 307C. It's the suite of offices that's almost directly across from 306, which is the usual general biology lab. And it's a suite of offices, so you just walk in that door, and my office is actually the one that's at the very end of the hall. Okay. If there's no more questions, I'm going to stop there. It's been a tough semester for all of us with all this COVID stuff, but there's some light at the end of the tunnel now. So if there's no questions, I will see you guys, remember, Monday, the 3rd at 10 a.m. in this room for the final exam. All right, have a good weekend, folks. I'll see you Monday. Okay, sure. Correct. Okay. And you said A was your answer? Yeah, I thought A was G. I start with A today. Oh, okay. All right. I'll tell you what, I will have to go in and correct everything straight.
I didn't want to study it wrong.
Yeah. Well, you got more points. Yeah, every now and then I'll. So it's question number 6. All right. I'll have to go back and fix that. Thank you for following up. Oh, your test. Yes.
Okay. Jacob, do we take the food donations to the building?
Refer to the flyer that I posted, because Biology Club is doing it differently this year, and I don't want to give you wrong information. Just go by what they're telling.
But do they take our names there and give them to?
Sorry?
Do they take our names there? And
Yeah, and if they don't, make sure you record your name somewhere. Even if it's like, I mean, a lot of students put their name down on a list for another one of my classes because for some reason this one wasn't listed. So make sure you report it somewhere.
Thank you.
You're welcome. And you're asking about the practical? Okay, I'll tell you what, as soon as we get back to the office, I'll get it online. Is it okay if we do it today? I don't think you'll have any problem with it personally. Yeah, I'm gonna have to.
Quiz, like how you like posted pictures there.
It's gonna be exactly like that. And I mean, you didn't have any problem with the other one. Yeah, what I'm gonna have to do is send you a password. Just so you know just so everybody can't take it. All right, yeah, I'll take care of that when you get back to the office.
Thank you.
You're welcome. Oh, there's been a lot of that.